Everyone, welcome. We're going to have a lot of fun tonight. This should be really special and really great, and I'm so glad you're here. I'm going to, in a little while, introduce the speaker, but this is a special event tonight, as I think most of you know, jointly sponsored by Carnegie and by the Norwegian Embassy and the Kavli Foundation, everybody working together to bring you a very special time. So my job right now is to introduce to you the ambassador from Norway to the United States, Kari Os.
Thank you, Matt. Dr. Scott, Dr. Raichle, Dr. Sejersted, Dr. Mi Young, distinguished guests. It's an honor to be a guest at this prestigious institution and in such esteemed company. The Embassy has had a long and fruitful relationship with the Carnegie Institution for Science here in Washington, D.C. As I look around the room, I notice an increasing number of attendees at this event every year. This is fantastic, as it must mean a need is being filled. In 2005, the Carnegie Institution welcomed Their Majesties King Harald and Queen Sonja of Norway as part of their official visit to the United States. I also attended this event back in 2005, and I clearly remember the High North and the Arctic as important topics being discussed. 10 years later, this issue has become even more important. This is evident as world leaders gather in Paris this week to discuss climate change. The Arctic region is an area in the world that is experiencing the strongest impact of climate change. But our relationship with Carnegie goes even further back than that. From 1906 to 1945, the Carnegie Institution supported the research of Norwegian physicist Wilhelm Bjerknes, who later became known as the father of modern meteorology. Tonight, we celebrate the Kavli Prize, a partnership between the Norwegian Academy of Science and Letters, the Kavli Foundation here in the United States, and the Norwegian Ministry of Education and Research. It is my privilege to be here tonight to express my sincere appreciation for the long-standing collaboration between Norwegian institutions and the Carnegie Institution for Science. The Kavli Foundation is the legacy of Fred Kavli, a remarkable man with great visions and strong dedication to science. Starting with an engineer degree from Trondheim, Norway, he built a highly successful business in California. When Fred Kavli eventually sold the company in 2000, he set aside a substantial amount to support science for the benefit of humanity. The Kavli Prize has been awarded since 2008 in 3 different categories: astrophysics, nanotechnology, and neuroscience. This year, we have the privilege to hearing from 2014 Kavli Prize winner in neuroscience, Dr. Marcus Raichle of the Washington University School of Medicine in St. Louis. I had the pleasure of meeting Dr. Raichle last fall when we, along with the other Kavli laureates, visited President Obama at the White House. A short time after that, the Kavli laureates traveled to Norway to attend the medal ceremony in Oslo, presented by His Majesty the King. I must admit, science is not my best subject. I was a little confused last year when we met Dr. Raichle, when you tried to explain to me the details about your impressive work and contributions. Although I can't say that I'm any less confused today, I do believe that your lecture this evening on the restless brain will provide some clarification, and I'm eagerly looking forward to it. Dear guests, it's now my pleasure to invite Professor Ole Sejersted to the podium. He is the Vice President of the Norwegian Academy of Science and Letters. Sir, the floor is yours.
Dear President Scott, Ambassador Aas, Executive Vice President of Science and Programs, Chun, Professor Raichle, ladies and gentlemen, it's a great honor for the Norwegian Academy of Science and Letters to team up with the Carnegie Institution for Science, the Norwegian Embassy, and the Kavli Foundation for the 7th Kavli Prize Laureate Lecture here in Washington, D.C. The lecture is an annual event and has become a tradition the Academy appreciates highly. This year's lecturer is Professor Marcus Raichle, the Kavli Prize Laureate in Neuroscience in 2014. Dr. Raichle received the prize with Brenda Milner and John O'Keefe for the discovery of specialized brain networks for memory and cognition. Reichle's observation of systematic patterns of ongoing brain activity when the subject is in a resting state has transformed the way the human brain is now being studied in health and disease. The Kavli Prize is a partnership between the Kavli Foundation, the Norwegian Ministry of Education and Research, and the Norwegian Academy of Science and Letters. The Kavli Foundation and Kavli Prize were established by the Norwegian-born entrepreneur, business leader, innovator, and philanthropist Fred Kavli, as the ambassador mentioned. The Kavli prizes are awarded for extraordinary achievements in 3 research areas: astrophysics, nanoscience, and neuroscience. And the purpose of the prize is to recognize outstanding scientific research, to honor highly creative scientists, to promote public understanding of scientists and their work, and to foster international cooperation among scientists. The Kavli Prize is awarded biannually in Oslo. The first time was in 2008. The next time will be in September 2016. The prize is awarded by the King of Norway, as you can see here. For each of the 3 Kavli prizes, astrophysics, nanoscience, and neuroscience, a prize committee is appointed by the Norwegian Academy of Science and Letters. Members of the committees are world-leading scientists within their fields. The committee members are chosen by the Royal Society of the UK, the French Academy of Sciences, the Max Planck Society, the National Academy of Sciences in the US, and the Chinese Academy of Science. It is the work done by the committee members that has given the Kavli Prize its high reputation. The Kavli Prize Laureate Lectures have become an important part of the Kavli Prize activities. This year, there were Laureate Lectures held in Berlin, Tokyo, and now in Washington, D.C. Today, we are here in the Carnegie Institution for Science to honor Professor Marcus Raichle. I look very much forward to hearing your lecture, but first I ask Dr. Myung Chun, Executive Vice President of Science Programs at the Kavli Foundation, to come to the podium.
Good evening. Thank you very much for coming tonight, and what a turnout. This is wonderful. First, thank Dr. Mascott to host this event in such a beautiful lecture hall. And I would also like to thank Ambassador Aas and Dr. Sugesti for giving great description of the Kavli prizes. I thought that I'll spend the next few minutes to share Fred Kavli's passion for philanthropy and how his passion really transpired into our programs, such as the Kavli prizes. The foundation's mission is quite simple, and it can be categorized in 2. One is to advance science for the benefit of humanity, and the other is to promote public understanding of scientific research. And it's in that endeavor that we have our Kavli Prizes program. Since it was already mentioned a lot about the details of Kavli Prizes, I thought I'll spend next few minutes saying a few words about our effort in advancing science through Kavli Institutes. And in our minds, it all began with Andrew Carnegie and his spirit. This is a picture of Andrew Carnegie, who's on the left, standing next to George Hale, who's a really well-known astronomer. In front of the Hale telescope in Mount Wilson out in Pasadena. It's about 1.5 meter telescope at that time. In 1908, this was the largest telescope. When Hale went to Andrew Carnegie and asked for and soliciting for funds to build this telescope, there was only one thing that Andrew Carnegie asked, and that was for this telescope to be possibly used by public at some time. And in effect, about 100 years later now, it's been 90% of this telescope is being used for public's outreach program. It's this concept of philanthropy being the private initiatives for public good without any demand. This concept was very inspiring to Fred Kavli, and it shows in our programs. And one of the most important programs at the foundation is Kavli Institutes. This is the way we support research. There are 20 Kavli Institutes around the world, 7 neuroscience institutes, 5 nanoscience institutes, 6 astrophysics, and 2 theoretical physics institutes. Fred Kavli believed that science has no borders, and so we have institutes around US as well as Europe and Asia. The mission of these institutes are to expand the boundaries of human knowledge through financial endowment that provides annual revenue. So these are institutes that receives funding every year forever. It's not an institute where you have to apply every 3 years or 10 years. It's a full commitment. It, I think, flavors very much like Carnegie Institution for Science. Our funding is unrestricted, and our only expectation is for our scientists to really take risks to address the best and bright ideas. And we believe that this will be the true discovery engine. And in many different ways, this is a very old-fashioned philanthropy. Today, people talk so much about venture philanthropy and milestone-driven research and so on. And we believe that Andrew Carnegie's way of providing endowment and having scientists' ideas really taking chances and going forward for long term is what we believe in. Because of our Kavli Institutes and our scientists who are in the field of nanoscience, neuroscience, and astrophysics, we're very cognizant from the foundation of selecting Kavli Prize winners be very independent activities from the foundation. So I wanted to just emphasize that all the activities that relate to selecting Kavli Prize winners are the responsibilities of Norwegian Academy of Science and Letters, great partners of our foundation. The foundation's role for the prizes program is for us to provide cash reward of $1 million per prize and gold medals. I have to confess that Fred Kavli worked really hard in designing this gold medal, along with our Kavli Prize program officer who's sitting here, Nadine Stern. They worked days and ends and so on. And so those are the kinds of activities that we have supported, but not when it comes to selecting the winners. When it comes to winning some awards, Fred Kavli has done not so badly. After all, he won the Carnegie Medal of Philanthropy in 2011, which he was really excited. In his speech, he mentioned that this is something that he says all the time. And those of us who have known him, he was such a visionary and in a very long-term visionary. So he would like to say, I like to look into the future. I think it's important for the benefit of all human beings. And it's his spirit that we think is very important for advancing science. I wanted to close by showing this picture. I think that Dr. Stefsky shown a lot of pictures of what happened in Norway, but I understood that only 2 prizes winners, Nobel Prize and the Kavli Prize winners, are invited to the Oval Office. And here is an example of American winners of 2014 Kavli Prize winners shown here, including is Marcus Raichle. So thank you, Marcus, for giving a lecture today, and good luck. Now I introduce Dr. Matt Scott. Thank you.
Well, thank you for that wonderful bit of history. Of course, we love the fact that you emphasized Carnegie's influence on so many others. He was really a remarkable man. Many of you know that he donated 2,400 libraries all over the world. You think of the hundreds of millions of people who have benefited from that gift. Really amazing. And those medals of philanthropy are designed to continue this tradition of encouragement of people to do as he believed you should do, which is if you have wealth, you should give it away while you're alive, so you have some influence of what happens to it and direct it in good directions. No, he didn't believe in inherited wealth, so his daughter may not have completely agreed with him, but she was all right. So getting onto the, I also wanted to say, it's been fantastic for us to have this collaboration with Norway and with the Kavli Foundation, and we look forward to many more years of doing this special night together. So now let's turn to the topic of the evening, and I'll tell you a little bit about that. In the late 1800s, the Russian physiologist Ivan Pavlov noticed in his research on the digestive system of dogs that the dogs began to salivate as soon as they saw the white coats of the lab assistants who fed them. I once knew a dog like that. It was kind of scary, but however, this was not that kind of lust. The dogs didn't need to see or smell the food that the assistants brought. The white coats and later sounds were enough to stimulate a response. Here in Pavlov's dogs was a psychological principle, the conditional reflex, that also applied to humans. Just as importantly, it was a principle that could be studied objectively. Psychology was on its way to becoming a science. Pavlov's experiments on conditioning set the stage for a branch of psychology called behaviorism that was to dominate the first half of the 20th century in America. Behaviorists thought that our behaviors are shaped solely by our responses to external stimuli, and that internal mental states, such as thinking and emotions, play little roles. John B. Watson, who coined the term behaviorism in 1913, actually felt it was impossible to study consciousness, and that to be a real science, psychology could focus only on physical responses. While Pavlov has often been called the grandfather of behaviorism, he did not think, like Watson, that human consciousness was unimportant. Quote, "It would be stupid to reject the subjective world," unquote, he said in his later years. Quote, "Our actions, all forms of social and personal life, are formed on this basis. The question is how to analyze this subjective world," unquote. It took many years before psychologists could return to this question. When they did, they brought with them a new focus on neural-based explanations of mind and, notably, an arsenal of brain imaging tools, such as MRIs, magnetic resonance, that could measure brain activity and metabolism and their relation. Despite this, behaviorism continued to inform neurological and psychological experiments. Brain activity was measured mostly while participants performed specific tasks or in response to external stimulation. The resting brain was considered hardly at all. That largely changed, however, in 2001. In that year, Marcus Raichle, our guest tonight, published a seminal paper describing the resting brain. He and his colleagues found that when people do nothing but daydream, several areas of the brain actually increase in activity, chattering away at each other in a highly organized manner. When engaged in a goal-oriented task, say when a daydreaming person reaches for a glass of water, these regions suspend their activities. Because this neural network turns on when we are not doing anything, when we simply introspect, Dr. Raichle called it the default mode network. This is like you sitting here thinking you're not doing much of anything, when in fact you're whirling around a huge globe spinning at hundreds of miles per hour. Try Dramamine. The default mode network that he recognized seemingly represents the brain's dominant intrinsic activity. Its discovery has been called a paradigm shift in brain research. And while there is still much debate about what the cortical associations of the network mean, today there is hardly an area of research into our health and our neurologic diseases in which it has not been implicated. Dr. Raichle is a professor of radiology, neurology, and biomedical engineering at the Washington University School of Medicine in St. Louis. He received his bachelor's and medical degrees from the University of Washington in Seattle, where he still has a home, and joined Washington University in 1971 after doing internship and residency programs in Baltimore and working as a neurologist and instructor at Cornell and the University of Texas. He also served for 2 years in the U.S. Air Force as a neurologist and flight surgeon. Many kinds of experience contributing to the perspective of a scientist. For many years after becoming a professor at Washington University, he also served as a neurologist at St. Louis Barnes Jewish Hospital and at the St. Louis Children's Hospital. Dr. Raichle and his colleagues have made pioneering contributions to cognitive neuroscience through the development and use of imaging techniques, including positron emission tomography and functional magnetic resonance imaging, or MRIs. A landmark study he published in 1988, representing 17 years' worth of work, described the first integrated strategy for the design, execution, and interpretation of functional brain images. Another of his studies led to the discovery that blood flow and glucose use change more than oxygen consumption does in the active brain. He is a member of the National Academy of Sciences and the Institute of Medicine, and a fellow of the American Academy of Arts and Sciences and the American Association for the Advancement of Science. He has received dozens of prizes and awards, including the Bristol-Myers Squibb Award for Distinguished Achievement in Neuroscience Research, the Grauemeyer Award for Psychology, the Ralph Gerard Prize from the Society for Neuroscience, the George Miller Prize from the Cognitive Neuroscience Society, and the Ariens Kappers Medal from the Royal Netherlands Academy of Sciences. His latest award, as you've heard, for which we honor him tonight, was a 2014 Kavli Prize in Neuroscience. He received this prize in Oslo just 3 months ago, along with Brenda Milner and John O'Keefe, as you heard, who were honored for their discoveries of other specialized brain regions. The Kavli Foundation is dedicated to advancing science for the benefit of humanity, promoting public understanding of scientific research and supporting scientists and their work. This is, of course, what the Carnegie Institution strives to do as well. So we are pleased to join the Kavli Foundation, along with the Royal Embassy of Norway and the Norwegian Academy of Science and Letters, to recognize Dr. Raichel's pioneering achievements. So please join me in welcoming a world-renowned neuroscientist who is helping us understand our subjective internal worlds and how they make us who we are. Welcome.
It's really difficult to follow such a set of introductions, but it's very humbling, I must say. but to our distinguished guests, the Norwegian Academy, the Ministry of Education, the Kavli Foundation, and now the Carnegie Institution. Thank you for having me here. It's a real pleasure. I'm also pleased to know that I have a second chance with the Norwegian ambassador to explain what I do. I hope it doesn't come to third time's the charm. Anyway, let me tell you, I want to take you through some of the things that have led me to the things I mentioned and give you a little bit of a sense of how my colleagues and I are thinking about this. For some of you who are ironclad neuroscientists, I will skip over a lot of details because I really would like to achieve some understanding of what we're up to, which has gotten ever more complex as the days, weeks, and years have passed. So I want to talk about the restless brain. And I think it's helpful for those of you in the audience who haven't thought about the tools that largely dominate what I work with. I should at least mention them up in front. We really had a revolution in the way we handle medical problems as well as how we view various organs of the body when in the early 1970s, X-ray CT, the CAT scan it was known as, was invented by Sir Godfrey Hounsfield in England. And this was utterly a phenomenal step forward in allowing us, certainly in terms of the brain, of looking at it for the first time without it dangerous, uncomfortable, and invasive approaches, which I learned as a neurologist. But the thing that it also spawned was a whole series of thoughts about how else you could image the brain. And I had gone to St. Louis as a neurologist to join a physicist, computer science people, and engineers using cyclotron-produced isotopes. And when this CT was first announced, their reaction, the engineering people, was, well, we can build a better CT scanner than Godfrey Hounsfield. And then along the way, it occurred to them that you could use these very unique isotopes to essentially take what neuroscientists had called tissue autoradiography, where you administer a radioisotope tagging some interesting compound, let it enter the brain, and then you remove the brain and slice it up, put it on a piece of X-ray film, and got a picture of what was going on. All of a sudden, we could do this electronically. Which was quite a step forward. So this is an incredibly early PET scan, but the map here is very different than that one in the sense that it delineates areas of the brain that have higher rates of metabolism, glucose in this case. But PET was not the only player in the field. Again, almost simultaneously, MR, which was nuclear magnetic resonance, which was converted to magnetic resonance imaging to get rid of the nuclear in the name. Also entered the field. And there you had these initially, and it's continued to this day, these gorgeous anatomical images of the brain. It was as if you could just reach out and touch the thing. But harbored within that technique was the potential for much more to be achieved. And I will explore that as we go along, but I will use interchangeably these techniques. But I'm also going to try to weave a story here that brings in other branches of science, because as we look at the brain from the top down almost, we are deeply dependent upon the wisdom of people studying the brain from the bottom up. And a plea that I often make is that we need to meet in the middle here somewhere and form a dialogue that will help inform us. And so I'll bring in the neurophysiology, the cell biology, and genetics along the way. And again, for you that are in those areas, it will be superficial, but I'd like to give you the sense of how these things might fit into the story. So let me begin a little bit at the beginning of this story. Why is it, for example, that we, with imaging devices that can measure simple things like blood flow and oxygen consumption and so forth, why would we even have suspected we could get anywhere doing this? Well, this is a very old story, and it begins with a fabulous scientist, Angelo Masso, who was a very eclectic Italian scientist who studied everything from high-altitude physiology to archaeology to the brain. But he had access to 3 individuals who had, for one reason or another, bony defects in the skull. This poor gentleman had been in a construction site in Italy and a brick fell on his head. He was patched up, he looked fine, but you could see the pulsations of the brain, just as you can see the pulsations in a young child through the fontanelles. And so he was recording this along with many other people, primarily in Europe, and he could record the blood pressure and he could record the pulsations over the brain. And on an occasion, it was actually Monday noon, September 23rd, 1878, and he was doing this recording, and the clock in the room struck 12, and there was some church bells in the background, and all of a sudden the pulsations over the brain rose and went back down again. So as a superb scientist, this didn't go unnoticed. So Maso said, he asked this fellow Bertino, he said, should you have said the Ave Maria? Because it was tradition in that Catholic society that saying a prayer when the church bells ring was the appropriate thing to do. And the fellow confessed that, of course, it would have been difficult being in the lab with this gadget on his head. But nevertheless, what was observed was this increase in pulsations once again. So Maso then did what I think is one of the most marvelous, it's essentially the first cognitive neuroscience study ever done. He asked the man to multiply 8 by 12. And when he was given the chore, up went the pulsations over the brain, and when he gave the answer, they went up again. And he basically said, look, the brain's blood flow is moving with the brain's activity, which is a fundamental part of the story that I want to tell. This is a marvelous story. It was lost to the dustbin of history until Gordon Shepard and I recently translated and published this book. And if you're interested in the history of science, I strongly recommend, not because we translated it, but because it's a fabulously interesting story of all the things he did. Well, in the modern world then, if we take PET imaging as a way to look at this, if you give somebody a checkerboard to look at, which is like a sledgehammer to the visual system, there's nothing sophisticated about this, you get a very strong increase in blood flow in the visual cortex, and you get a very strong increase in glucose consumption. But much to our complete surprise, this kind of was not the dogma. The oxygen consumption barely changed. We survived publishing this, but when you put something out there that doesn't fit, it is really a remarkable experience. We think of ourselves as being, open-minded as scientists and so forth, but you get very attached to the way the world is as you think about it. And many people have experienced that. But this turned out to be very important in a conceptual sense, and the story actually begins again with Michael Faraday. As you know, was one of the really great scientists of any time, who was famous for his work with electrochemistry and electromagnetism. And along the way, in his voluminous notebooks, he looked at blood, because as you know, blood contains iron, and it should react in a magnetic field in one way or another. And he didn't see anything. And he wrote in his book that should try liquid blood. And it was Linus Pauling, and how in the world he found that sentence in Faraday's thing, but he did it. He found it, and in 1937, the year I entered this world, he published a seminal paper that said when blood becomes deoxygenated, it becomes paramagnetic. That is, it disturbs a magnetic field. And it was this fellow who knew about Pauling and happened to know about our Seiji Ogawa, a good friend of mine, working at the Bell Labs with David Tank and others, who said, wait a minute, maybe we could use MR to make this measurement. And it's a very simple experiment. You imagine this is a little red blood cell, and it comes through, it loses its oxygen to the tissue. The magnetic field gets disturbed, and if you look at this little rodent he had, you can see those black lines, those are veins. So that's deoxygenated blood. And so he did the ingenious thing. He just put the little fellow on 100% oxygen. The veins disappeared, and he called it blood oxygen level dependent contrast, or the bold signal of fMRI. So this has become an utter workhorse for the kind of thing that we and others do. It's seen in things like this, where you have various tasks and states, and you subtract one thing from another. It's been everywhere from my cane's there. I don't need it at the moment. If I fall, I'll reach for it. So anyway, this kind of gives you a sense of what we're up to here. So now we're getting a bit more sophisticated in terms of the machinery we can use. This is a composite from a PET and an MR scan, which are now integrated into single machines. So you've got the body seen by the MR scanner and the soft tissue, and then you see the brain and a bit of the heart. But the impression I want to leave you with, and you're going to see this repeatedly, this is a completely normal individual lying in a scanner doing absolutely nothing that we could perceive. And you can see the level of activity in that brain. And just to put some numbers on it, the brain is 20% of the body's weight, or 2% of the body's weight, but consumes 60% of its energy ongoing. So it's a deeply expensive organ, even when you're not, if you will, using it consciously. And something that I'll mention very briefly at the end, but important to keep in mind, that in a child of age 10, it's 6% of the body weight. And in that state, where it's twice the metabolic rate, it is now 50% of the cost of running a 10-year-old child's body. And I think this has major implications just in how you treat it, if nothing else. That is, you need to protect it. So, how do I frame the question on the table in terms of intrinsic ongoing activity versus those things that are evoked in my brain as I give this lecture, or hopefully as you watch it? And again, it goes back historically to two general views of how the brain works. One, in a way, was certainly associated with Sir Charles Sherrington, one of the great neuroscientists, again. And his view, simply cast, was that it is reflexive, that the brain is sitting there waiting for something to happen, and when somebody in a white coat or your neighbor walks up, something gets lit in your brain, something gets turned on. These are words that are... There was an opposing view here by one of his students, T. Graham Brown, who said, wait a minute, this is really intrinsic, that it has planned for the future, it's got all these programs, and they're ongoing and manifesting in this activity. Well, the problem was, how do you decide who's right here? Whereas here, you can do all these experiments and ask your colleague or your laboratory animal or your cell in the tissue, you can play with it by putting in a stimulus and see what happens. But how do you interrogate, how do you get at it if it's not that? Well, the first thing is, should you waste your time at this? How do we adjudicate these two views? And I would put it in two rather simple terms. One is cost, and the other is connectivity. And by that I mean, how much of our brain, what of the world that I'm looking at and you're looking at me, does my brain actually see? So let me explain here. I've used this many times, but I think it gets the point across. Here you have images of a group of individuals, and each image is from the same group of individuals, and in each image they're doing something very different. And I honest to goodness defy you to see the difference. It's just not apparent. But we're reasonably clever, and we can subtract and average and all the clever things that have been developed over time, and you can see that very clearly you're processing words in various ways here, and we can delineate those areas of the brain that are associated with that. And we've seen this over and over again, and it's incredibly useful to do. But just notice the difference in the the difference is remarkably small. It's a few percentage of what's going on. So from a cost point of view, what's going on intrinsically in the brain has got to be important just for that reason alone. But the other thing is that as we think about how we view the world, we have this intuitive feel as if the world is playing out on a screen within our brain. Well, you can quite interestingly ask the question, how much of what's out there is actually getting into my brain? And so you can start with the visual system, and you get at the back of your eye, your retina, you get about 10 to the 10th bits per second. But you've only got a million fibers here, a million wires, if you want to think of it that way. And we know exactly how many bits of information each of those wires can transmit. So we know when it gets to the first way station, it's down 4 orders of magnitude. That's a big number. And when it gets to the visual cortex, it is down 6 orders of magnitude. That is an even bigger number. So what it says is that what is extracted from the environment is deeply compressed by the time your brain gets it. And the only inference one can draw from that is your brain has to apply some sort of algorithmic approach to reconstruct what is in detail out there. And the final thing to say is that I think we treasure our conscious, intuitive feeling about what we see, but keeping in mind that your consciousness is less than 100 bits per second of information processing. So there's not only a whale of a lot of stuff going on in there, but most of it is outside of our conscious awareness. So when you think about brain and behavior, we need to approach it with at least some of this in mind. Bernard Mountcastle recently died, but again, another great neuroscientist, and a very, very sober man. I knew him a bit. Not one to make wild statements, but his comment is straight on here. He said, each of us believes himself to live directly within the world that surrounds him, to sense the objective and events precisely, and to live in a real and current time. I assert that these are perceptual illusions. Sensation is an abstraction, not a replication of the real world. So, how do we, if intrinsic activity is this important, now we got a problem, because how do we study this with because it doesn't fit with the general paradigms. And what I would say, and what I want to share with you pretty much for the rest of this talk, is the serendipity and luck that led to us, and when I say us, many around the world, a better shot at this. For us, the first thing that came to light has already been mentioned. If you take you do a task, so you have a task here and you have some kind of a resting state here, and you look at the difference, you will see changes. It depends on what you're doing. And utterly by accident, just because we happen to collect what's known as a resting state, you put you in a scanner, you just lay in there and we take a picture. But it didn't fit into the general paradigm of what we were doing, of deeply controlled behaviors beginning with that control condition. But we just, by habit, collected it. And in a moment that I cannot actually remember, we looked at what happened in that, as you compared this just laying there with the task, and we noticed that certain areas of the brain decreased their activity. And of course, the immediate reaction was, well, you didn't control what they were doing. Heaven only knows what you were thinking. Well, this turns out that hundreds and hundreds of people must be thinking the same thing. And as was mentioned, it just kind of as a bit of whimsy, the paper that ultimately defended the biology behind this, and I won't burden you with that, it was published in 2001, but just as kind of whimsy, I titled the paper A Default Mode of Brain Function, and my good friend Mike Grecius tagged this network as the brain's default mode network. And this has received, as again was mentioned, a tremendous amount of attention. These are the now close to 6,000 papers that have actually spoken about this. And it ranges across a variety of things. And I don't plan to get into the details of what has been associated with. I think the list of what it hasn't been associated with would be much shorter. But the story would, I think, be laboring if some other serendipitous things hadn't happened. And one of them comes from the fact that's going to be illustrated here. This is an MR scan of a former graduate student of mine. So each row here is 1 minute worth of images of his brain. So there's 5 minutes worth of data here. And if you kind of look at this, you can see that it seems to change. It isn't exactly constant over time. So if you actually pick a spot, say over the motor cortex, and you look at, you see a signal that looks like that. Well, any decent scientist would have said that's noise. And the way you deal with noise is to just average it out. You get rid of it. But a physicist, graduate student, Bharat Biswal, at the Medical College of Wisconsin, his mentor, Jim Hyde, said, we need to know more about this noise. And the clever thing he did was to say, well, look, I've got this signal. Is it correlated with anything? And lo and behold, there was the whole motor system. And again, there was all kinds of controversy about, bad experiments and noise and so forth. But this was really a seminal observation, and it led to a whole field of doing just that, but with all kinds of sophistication. And again, I'm not going to get into all the ways you do this and the technical issues that are involved, but just to give you the gestalt, that if you go into the brain, you can pretty much delineate the major component of the systems involved. And so the default mode network is here, control systems, attention systems, auditory, sensory, motor. This was, honest to God, it was the darndest thing I'd ever seen. I mean, it was just surprising as heck to see this thing laid out there. But the first thing that comes to mind is, well, okay, wait a minute here. These are blood flows. Remember Maso and blood flow and metabolism? Neurobiology is spikes and field potentials and stuff like that. How do they relate? Well, there's been, again, a good bit of progress on this, and I think it opens up a lot of very, very fascinating things. We're not entirely ignorant about what is behind this, but we have much more to learn. So one thing that's been done is either to record the human electrical brain activity either by electrodes on the head or electrodes over the surface of the brain, and usually in patients undergoing epilepsy surgery. And if you do that, this is what you get. This was the stuff that Berger saw in 1929. And if you can get clever with this, and you can break this down into various frequencies. So you may have heard of alpha and gamma and so forth, maybe not. But this is the name of the game, and this is how you go about working with this kind of thing. And so the question then was taken at this level, is there any connection with that kind of electrical activity and the ongoing activity here that I just showed you, this stuff? And I would say the weight of evidence at the present time would suggest that it's related to what we call infraslow fluctuations. These are it's like ground swells, if you will, in the ocean. In fact, let me go back here. That probably are instrumental in setting the excitability of the system. So in one part of their phase, when they're increasing, cells become more excitable, and then they decrease in excitability. I think there's a plausible argument to be made that that is a very important connection, again, which we have to learn more about. So, okay, you got it tagged to one frequency, but there's a heck of a lot else electrically going on in that EEG, and if you put an electrode in, you see neurons spiking and so forth. How do you put all this together? And it resides around the fact that it's not a free-for-all in there. There is a set of relationships among these different frequencies in space and time that I'm going to explore a fair amount here for the next little bit. But in essence, what you've got here, imagine that this is your fluctuating resting state bold signal. But it's depicted like this, so you've got the phase of this thing, that it goes up and then it comes back down. And if you look at where these other frequencies reside in relationship to that, they're piled one on top of the other. They're coordinated across time. This is called cross-frequency phase amplitude coupling, but I think you get the sense of it is that these folks in there, these voices of the brain, if you want to think about it, that are speaking in different frequencies and so forth, are not doing this independently. And I often kind of muse about this in terms of if this metaphor may help you. And that is, think of the ground swells, and then look at the whitecaps. And when you never have a whitecap in a trough. It's always dependent upon the underlying activity of the system, which I think is an important way to open the door. So I would say at this juncture that the brain is intrinsically organized in both space and time. So how do we think about the evoked response? It's as though there's been a paradigm shift. We're not going to ask anybody to do anything. We're just going to look at what's going on internally. Is there any relationship here? And again, I think this is a complicated subject, but there are a few things that if you're going to speculate, you need to know. One is this. This was a wonderful experiment done by David Heeger at NYU and his colleagues a number of years ago. But imagine now you're in a scanner in David's lab, and you're asked to push a button when you see this very, very faint grating up there. And if you don't see it, you don't push it. If you see it, you push it. And so the critical thing is you're given a cue. You're told it's going to happen, and it does. And you push the button. And what you get if you look at the visual cortex is an absolutely classical, bold fMRI response. But every so often, you come along and you get the cue, so you're told it's coming, you're ready for it, it doesn't show up on the scene. And guess what? You get the bloody well same response out of the visual cortex. Now this, there's some explaining to do here. Well, this is not just an fMRI curiosity. This is a well-known phenomenon. This is a very famous paper by Kenneth the People de Weitzman, done in 2003. It was a Nature paper. So if you present a particular type of visual stimulus to a cat, you get a response using optical imaging techniques now, various bands and so forth that have a very characteristic pattern. But what's extremely interesting here is this is an evoked one, this is an average one. This is the spontaneous activity of the system. And what it's doing is continuously revisiting what it's likely to be seeing. So, my one-sentence interpretation of this is that our brains are seriously in the prediction business. And that for the most part, that dominates a good bit of our behaviors. We can probably suspend that. We can think fast and slow, as Danny Kahneman said, but I thinking fast is a big part of it. And all of this is done in a very interesting and automatic way. Now, we've made a little progress here, we, the large community. But if you look at a picture like this, and you see these marvelous little these networks in here, and you can make more or less of them, you can do all kinds of fancy things. It looks like a bunch of balkanized states. They're not talking to each other, and yet I know for a fact that I'm talking to you, I'm moving this thing in my hand, and if my visual cortex is not talking to my motor cortex, there's something wrong here. So where does that come from? How do we think about how we have this within a system integration, but we have to integrate across systems? How does the brain talk to itself around its components? So, what is it about time? How does it fit into this picture? This was something that I know a lot of people had thought about, and it was very tempting to take movies of resting state MR and sort them according to images, to networks, and by God, you'd see stuff moving around in there. But it turns out that was noise. I was very disappointed, actually. But then there's nothing like a great graduate student. It's the history of my life, great colleagues and great graduate students. Anish Mitra, he came to me as an MSTP student. He was an advanced math major at Stanford. He had worked at Google for a while. He was a concert bassist, and he was a member of the Stanford debating team. And he's the nicest young man I've ever known. But he got to thinking about this problem, and the question was, here's our signal. Okay, if you look at this, you just take an arrow here and an arrow here. And if you look at the signals coming out, they're sure as heck correlated. That's for darn sure. And if you just kind of stretch your imagination, you might imagine that one of those curves slightly leads the other one. But if you're not persuaded, I wouldn't be offended. But it turns out that there are and I will not get into this, but if you really want to dig, these are not the easy reads. But there is a way to mathematically look at where the center of that distribution is. And you can do this with incredible precision. And the data I'm going to show you involves 1,400 subjects. So it takes a lot of data to pull this out. But by God, there it was. So this is a completely different view of the brain. And what you're looking at here is a latency map. That is, areas that are active early are blue, and parts of the brain that are active late are red. So you can see that if I were to present you with a movie, you would see things moving across the brain. And it turns out there are multiple components of this, which we won't get into today. But they're, if you will, orthogonal to the resting state networks. They're going across rather as well as through them. So this is another picture of it. And what lies in behind this is an image of this that I must say was an acquired taste even for me. So, but here it is. So, now we're getting a little further along in this, but here's an attention system up here, our favorite friend, the default mode network here. And the networks themselves, that is what's going on and within them, are in this diagonal here. And you can see that in each one of those networks, some parts of it are early and some parts of it are late. So the default mode network tends to be early in the back and late in the front, and the attention system is the other way around. And you can go through all of this, but these relationships are also early and late with all their neighbors. So you have this incredibly complicated relationships going on in there, but it opens the opportunity to think even more in more detail about how the brain is actually working. And I have to say, I'm actually surprised how far we can take the imaging to get at this. It really has kind of amazed me. But so you might say, well, the picture isn't this, it's more like this. And again, all the mathematical wizardry that led to all of this and all the caveats that I'll leave you, the aficionados in this audience. But I just want to give you the sense that we have to think about this in a very sophisticated and integrated way. So what might the internal dialogue then look like here? If we've got all this going, you could say, oh gosh, it's just, you're not going to get anywhere with this. I'd like you to think about it a little this way. We've really struggled with a metaphor of how to think about this, because you have traffic moving in there at different frequencies and moving in different directions, but still maintaining a sense of systems within that. And what emerges as you think about this is a dialogue. And this is an idea that Yuri Basaki has talked a good bit about, and that is that in the course of the conversations, you have receivers, people that get the message, and the senders. And that can probably be related to the infraslow stuff. The receiver says, I'm ready, and the sender sends. And so just think about the possibilities of this. So now you come back to this thing, which I showed you earlier, cross-frequency phase. So here's all these different frequencies here, and they're being lined up by this internal underpinning of excitability, if you will. This, of course, is moving over time. And what we now begin to suspect that each of those frequencies is a voice of a different system. And so that you can imagine then a telephone conversation. So you have a telephone conversation, you listen to the person on the other end of the line, and generally if you know these people, you know who they are. And the conversation moves back and forth. So there are senders and receivers here. It's a simple metaphor. I don't want to trivialize what we're doing, but I was just trying to think, well, how do I get you up where I'm wrestling with all this? And of course, there's a more modern version of this. For those of you that don't know about picking up the phone and doing things of that sort. Well, enough of this. The one thing I want to finish up with a few final remarks is this whole business of cost. So however this is organized, and it appears ever more beautiful how it is actually organized, should be no surprise, but you have to come back to say, well, wait a minute, it is also deeply expensive. And how do we begin to assess that cost? We know for a fact that one of the signaling things in the brain, that is the very famous spike that neurons do, is incredibly cheap. A single spike is not very expensive, so you can't pin it on that. So we have been looking at this a great deal, and when you talk about cost, you'd say, well, what's the currency here? Well, the currency is glucose. For the brain, it's very finicky. It takes sugar, and it burns it up to carbon dioxide and water, and it produces a great amount of energy down here. But it goes through a whole set of steps, and I'm sure not too many of you are biochemists. And if you had a course, you probably, as I did for many years, didn't take it with you. But it suddenly became very important for us to understand what's going on. So this wonderful little molecule, glucose, pops into the brain and goes through a little step. And then if I were to ask a neuroscience audience what happens after that, they jump right down to here. Why? It just gets burned up in the furnace and produces all this energy. If I were to give this talk to an oncology group, a cancer biologist, you would get a completely different answer. They are, if you will, obsessed with that, what's going on there. And what this is called is glycolysis. And the question is, what the dickens is going on? This is a wildly oversimplified picture of it. Sorry. I keep looking over at the ambassador here, hoping that he hasn't checked out or is just looking disgusted. But anyway, what oncologists, this was Otto Warburg discovered this in 1929, that cancer cells, if you decide you're going to be a cancer cell, you shift your metabolism over to glycolysis simply because that's how you build things. It's very concerned with biosynthesis, a little bit concerned with energy that's highly placed. So the question is, does this have any connection with how this might be played out in the brain? And so you could say, well, what do we know about it? Well, first we know that if we look at the brain, big surprise to us, that certain areas of the brain, including the default mode network, are highly glycolytic in the adult brain. And areas like the cerebellum is off the charts in the other direction. And exactly why that is the case, we're not certain, particularly with regard to the cerebellum. As I showed you early on, this increases in areas where you have increased activity. It is deeply important in that 10-year-old infant whose brain is 50% of the cost of running the body. About 30 to 40% of the glucose use there is aerobic glycolysis, as we call it. Well, that fits rather nicely with the fact that you are at the peak of synaptic proliferation at that point in your brain development. And so that the question that then arose for us was, can we put any better data behind that? And this was a collaboration we had with the Allen Institute in Seattle and the BrainSpan data at Yale, where we asked the question, what sort of genes migrate with glycolysis in the human brain? And it turns out that areas that are very glycolytic are deeply concerned from a genetic point of view with plasticity, whereas other areas, I mean, it's not an either or, it's a balance. But the balance is more in favor of just synaptic activity. So these areas, then, we have to begin to ask ourselves, what is unique about it? Why is it using these metabolic pathways in the way it is? Because I think it will be deeply informative and also help us think about a variety of issues. This is just a quick piece about that child's brain, because I feel that it's rather important to realize, and this what bothers me deeply at times, is watching little 10-year-old kids with football helmets running around bumping their head. And I'm thinking, doesn't somebody realize that that's a precious piece of equipment in there, and it's at the peak of activity and doesn't need to be bumped around? I'll just leave it at that, but I think I've made my point. The final point I want to make with the metabolism thing is, and this was, again, one of these big surprises. As you know now, we can take a PET scanner, the community, and essentially look at the ravages of Alzheimer's disease in the brain, the amyloid plaques and the tau tangles. And when the people in Pittsburgh first put forth this, what we call Pittsburgh Compound B for PET, to image the amyloid plaques, what stood out, although it wasn't put in their first paper, because I was told in retrospect they thought it was an artifact, but it's indeed not an artifact. So you have the areas of high aerobic glycolysis, and just look at the distribution of the plaques. So the question comes up then at a more fundamental level, what exactly is going on in the Alzheimer's brain that leads a normal constituent of that brain, that is amyloid, that's part of the natural synaptic machinery of the brain, that increases when the brain is more active, decreases when it's less active. What causes that to precipitate? The answer is not in amyloid, the answer is in that which it is involved in terms of the synaptic physiology of the brain. So there are a variety of things out there that we can look at. For me, it's been an amazing adventure to have had to be at the table when all of this has unfolded. The brain's activities, as I've tried to make the point, are mainly intrinsic, ongoing, and costly, and importantly non-conscious. And that is a subject of immense importance, I think, as well as we think about human behavior and our understanding of why we do what we do and why we feel what we feel. I'm often taken by Mike Gazzaniga's great experiment called the interpreter brain, where you have a separated hemispheres in which you ask the right hemisphere, which has no language, to do something, and the left hemisphere, which has language, offers an explanation which bears no relationship to the behavior. We'll put that in the context of a brain in which the vast majority of its ongoing activity, anticipating your every need, is going on about business without your conscious awareness. So when somebody says, well, I like this red Ferrari rather than a blue Chevrolet, there's more to that answer than some sort of reasoned wisdom. I think this illustrates the point I've been dragging home here. The brain's like an iceberg, and the majority of it is that we can see is the minority of what's really, really there. But for us to get anywhere with this enterprise is that we've got to foster a dialogue among levels of analysis here. Nobody's got a corner on truth here, and we have an immensely interesting and challenging task before us. Again, it's been a real honor to do this. I just thank the Kavli Foundation, the Norwegian Academy, the Norwegian Ministry of Education, all the people that have flattered me beyond belief. And I'm grateful, and I say this for a large group of people that made it all possible for me. Thank you very much.
So we have microphones set up in both aisles, and feel free to step up to them and ask some questions. We'll do that for a little bit. That's great.
Thank you.
Dr. Raichle, I'm sorry, I have to tell an anecdote to get to my question. Anybody who can see my shoes knows I'm a bicyclist. Two-lane road, two two-lane roads intersecting in a T. I'm on the top bar, coming along in the dark with a very bright light. At the intersection, there's an SUV, very nice, stopped carefully, apparently looking both ways. As I'm passing, the SUV starts out. I pay no attention because I think that they're going to be on the left-hand side of the road and pass me and go ahead. About 10 seconds later, or no, 2 seconds, I realize that the SUV's front bumper is right here, 6 inches from my shoulder. No contact. I just continue on. I look at my mirror and I see the SUV 500 or 600 yards back. Apparently, the driver trying to get his or her heart started again. What is your best guess at why this person, who very probably saw me, at least the light photons got into the eyes, did not in fact see me? Is that what it looks to me like the prediction was not expecting to see a bicycle coming along, looking only for cars, and therefore didn't see a bicycle coming along. Do you think that's...
You frame an interesting situation. I don't have an exact answer to your thing, but you know, I often think about stuff like this. I've been driving the same freeway home for 44 years, now going on 45 years, so I know that freeway very well. And I get there and you're always thinking about a lot of other things, and yet your brain is driving this car, honest to God. And unless somebody swerves out of a lane, your mind goes along. And another test of it is that my wife says, well, you stop at Deerberg's to get X, you know, and you get all the way home and you didn't do it because it was not part of the plan. So, and the other thing that the anecdote, I'm a great fan of the essay, The Secret Life of Walter Mitty. Read that and think about this, because here's Walter, he's going along, he's in command of a ship, he's a great surgeon, he's doing all these things, and every so often, and he's driving a car. That's how it starts. And then all of a sudden, a voice breaks in, Walter, slow down. And you realize that what we mean by attention here, I think, is a much underappreciated issue. We think of it in terms of my goal-directed behavior. I'm in David Heger's experiment looking at a grating and I'm trying to push a button. But your brain doesn't just turn on for that. It is attending all the time. And I would like to emphasize one other thing, is that as people characterize, say, the default mode network or any of these networks, and they say, well, it's off when you're doing something or on, nothing is ever off. It's all this very, very gentle balancing of these relationships. And those things are utterly critical. But what goes through your mind, I mean, you could read Danny Kahneman's Thinking Fast and Slow. Obviously, whoever put the brakes on got it right. But there's much to your question that I puzzle about, and I don't have a great answer, but thank heavens you're here.
Yes, thank you.
Okay. Another survival story.
A little bit different take. I'm thinking of, I have a nephew who's now going on 13, but starting when he was very young, he was very physically active. He needed a lot of stimulus. And I remember talking with my sister-in-law about it, and she said, he's listening to you, but he needs to move while he's listening to you. But when he gets to something he wants to do, he can be very focused. I mean, he's very good at, he can sit down and write a paper, he can sit down and read a book, he'll be very quiet, he'll be very relaxed. But if you want him to kind of participate in things, then he requires a lot of stimulus to almost activate his ability to participate. And I was wondering if you might have any insight into why that might be in his case, whereas not in, say, his brother, who's 3 years younger, who's, I guess average.
The answer is no. I think maybe someday we will be able to approach these. And one of the things that's been very encouraging as MR moved into the picture is imaging young children, particularly newborns and premature infants and so forth, is being done routinely. And so our ability, there's the terrible twos, and the imagers, we haven't figured out how to image those kids, but then when they get, when they arrive at about age 6, they become starlets, and they get a big kick out of getting in the scanner and having their picture taken. But I think the thing is that trying to begin to understand how these things develop, as I kind of hinted at here, and I didn't really get into the nitty-gritty of all these things moving in different directions and all their components and so forth, but as the richness of this improves, I like to think that there is increasing room for mischief in there, that little things that are happening in these very complex relationships and that can be ascertained in the human brain to begin with will become valuable in understanding these things. So I'm optimistic that some of these really troublesome things about development, the whole issue of self-control, which we've been involved in, I think there's a way to be optimistic about this in the future, but we're a long ways from being there yet, with a lot of challenges technically. But I just hope to hang around a little longer and see what happens.
Thank you very much for this fascinating talk. I just wonder if you could say, with this default mode network and with the picture of the iceberg with consciousness being just above and much more being underneath and being defined by waves and responses, the earlier paradigm that replaces is somehow more appealing for the study of learning because it's more about tasks and responses and it's simpler. Could you say a few things about how this new paradigm might interact with or change the approach to understanding learning in the brain and the changes in the individual over a lifetime?
It's a great question, and it's something that you'd think you might ask the question, am I even focused? But there's many things going on around me. We had a relationship with Giulio Tononi for some period of time at the University of Wisconsin. Some of you may know him. But in the course of that, we were very interested in the basic issue of your question, and that is from both a metabolic and a functional connectivity, if you will, and a task activity point of view, can we make some progress in understanding what it is to learn something? And these data we've been wrestling with for about 5 years, but they're now finally in review. It was a multimodal experiment involving all these different modalities on the same bunch of people. And the paradigm was like the prism experiment, where you put on prism glasses and throw your something at a target and you miss, and very quickly you compensate. This was a motor adaptation, similar but with a cursor and so forth. And what was really striking about this is that in fact you could, through the task data itself, the task data was critical to this, we could isolate the components that the brain used to do the task. And from that, we could isolate a remarkably small number that were affected by the learning of the task. And then we could show that the relationship between this particular area in the frontal cortex and the visual cortex had changed its connectivity. And then we could couple that with the metabolism to show that the aerobic glycolysis had gone up and the oxygen consumption had gone down, which is would be predicted by something called LTD. And what it brought into the whole picture then, which has taken us so much time to come to grips with, is the whole issue of synaptic pruning in the brain. And the critical cell here is the microglial cell. That's the immune cell of the brain. Well, I mentioned Otto Warburg and the aerobic glycolysis, but it turns out your immune system uses the same trick. So your T cells, when they decide to attack something, they upregulate aerobic glycolysis. And guess what? The oligodendrocyte does the same thing. So the whole implication of the line of reasoning, which is hard to lay out in a few sentences here, is you can combine the task where you're going after a behavior and then look at the context in which that task has occurred, and then the before and after and the changes metabolically, which reflect something about the cell biology of what's going on. And a task like that, of course, now you could go back and take it down to other levels and look at it. But Rob Malenka, some of you may know him, he's one of the world's authorities in LTD, and one of the points he makes in one of his many reviews is that, well, much progress on something like the question you're asking has been made in simple systems where you have cells in a petri dish or a slice or something, that heretofore there had never been a demonstration that it was related to an in vivo human experience of learning. And so I'm kind of optimistic that we can pull off stuff like that.
Thank you. Thank you very much.
Yeah. What I wanted to sort of ask you to talk about is attention and training the brain for attention. And the reason I'm asking that is because to some degree we start out as that tabula rasa, which is, you know, sort of blank. I mean, we've got to maintain our physiological stuff and pain and all of that kind of stuff. But culturally and over time, the way we train the brain to pay attention to the infinite detail of the outside world, we don't really look at that. I mean, we have education systems, but I don't know whether we're really doing the best job of analyzing how we want to set the brain up for a lifetime so that it can manage the greatest amount of relevant information over a lifetime. And it seems to me that things can move so quickly that we can't keep up with it. In other words, we can't program ourselves to get the proper amount of attention to the things that we should be maybe as, and I'm just curious if you've looked into that at all.
The answer is no. Have I thought about it? Yeah. Truly one of my great colleagues in this whole thing was Mike Posner at the University of Oregon. He's one of the world's top cognitive psychologists. He was utterly instrumental in developing of image subtraction and cognitive subtraction with us many years ago. And he spent a good bit of his life with Mary Rothbard thinking about problems just like this. So he's the guy to answer a question like this. I must say I'm a big fan of NPR and NewsHour, but they had a piece on a while back about classroom activities, just the last week I think it was, where they had interjected all kinds of activities to accomplish what you're talking about, and they called it a brain-based approach. And I have to confess I had trouble with that. I really did, because I don't think we're in a position to contribute. I don't know where the ideas came from. These were highly energetic teachers. The kids looked like they were having a whale of a lot of fun. But how I would think about it from a brain science point of view, I just really couldn't. The other thing that's in there, which is a really interesting problem that people have thought about this for a long time, and that's the problem of mental effort. Think about it. If the classic task, and it was worked on by the British during the Second World War when they had people looking at the radar screens in England, and the question was, how long can you focus on a radar screen to see that incoming German fighter plane? And it turns out that you can't go much beyond 30 seconds. That may surprise you, but it becomes effortful. You just can't do it. And I've talked to Mike about this several times, and the issue is nobody quite understands why. Is it because it costs a lot? Well, I'm a cost guy, and I have a hard time with that. Is it because your brain has a certain momentum, and you can kind of, it's like a super tanker, you know, it kind of knows where it's going, and you can kind of pull it off course for a period of time. But how does that? And when you say effort, it's like, you know, my muscles. You know, you run a 100 yards dash and you're fatigued. It's not that simple. So the whole issue of how attention gets parsed here, I think, is a fantastically interesting question, and I don't think we really understand it very well.
Thank you very much. This has been fascinating. There are two topics that have made the news lately. One is sleep and the necessity for sleep and what that does for your brain, and I'm wondering if you've done any work in that area and have any insights. The second is the biota of the intestinal system apparently has quite a link to your brain, and that kind of raises the issue of nutrition and how that might also affect the system.
Do I have another hour?
Yes, yes.
And this will really sound unfocused. Yes, we have worked on those things. I'll deal with two of them. Studying sleep in an imaging device is extremely difficult because it's so unnatural and it's noisy. So getting people to go to sleep in an MR scanner ain't easy. But there have been a number of attempts, and if you get into this, you have to be seriously committed that most of your subjects are not going to accomplish this. But Helmut Laus in Germany, a good friend of mine, pulled this off several years ago, and he got a very large amount of very pristine data in slow wave sleep, which has been pegged as a very important event in the terms of kind of reorganizing my brain for the next day, if you will, in the simplest possible terms. And so this business of latency, and I haven't talked about lags, and I haven't talked about motifs, all the inner workings of this sort of thing, we had this data, and that was just published in eLife, that we looked at all of this. There are really remarkable changes going on in slow wave sleep. REM sleep seems to be just off the charts. Nobody can, because it's very brief. I have this watch, you know, these Basis watches. I'm getting old and I don't sleep much, and I wanted to know. And you see the REM episodes, but they're fleeting and they're in there and out. But the first order of business is there is a major reorganization in slow wave sleep that you come out of. But the other thing which we were wrestling with, which led to those slides on the conversation here, there is a conversation between your hippocampus, which is important for memory, and your cerebral cortex. So that in one instance, information comes, the sender is the cortex and the receiver is the hippocampus. In slow wave sleep, that flips. That was just a stunner. And this is what got us into thinking and how you envision the direction of traffic on these, is it the wires, is it the modulators, how does this happen? But it is unambiguous. And Yuri Basaki, among others, has talked about the potential for this, but a direct demonstration of it had never occurred. Now we can't, and we did it with electrocorticography in some epilepsy patients and in this cohort, and it's both electrically and fMRI-wise there. So yeah, you can approach this. I mean, we spend a lot of time sleeping. We spend less time sleeping as we get older, and one of the questions that might arise is the lack of sleep a risk factor for aging and neurodegenerative disease? And I think we have not explored that adequately. It's not easy to do, but I can tell you from personal experience and large epidemiological studies that as you get to my age, you don't get as much sleep. And so hopefully after this I'll get a good night's sleep. And then the issue of the intestinal biota, I'll just say briefly, a few of you in this audience may know about this, but we have one of the world's authorities on that in Jeff Gordon at Washington University. He's a friend of mine, and a few years ago he got one of his many awards, so I called him up and said, "Hey Jeff, you ever think about the brain?" And that has created a dialogue that is involving with other people now issues of nutrition, undernutrition, malnutrition, and in the premature infant where nutrition is a huge problem. And trying to understand how that influences the brain. And so you have both a human approach to this problem, and then there are these so-called notobiotic mice. So you start with a mouse with no bugs, and then you put different bugs in and look at it. So, you know, I think a number of things have been said about that paradigm. There's a good bit written about it, not just from him. I think we have a long ways to go. I think it's a bit reckless at this point to say, well, your intestinal biota might have something to do with whether you get schizophrenia or something. I think that's not responsible. But I think there is much to be done here. But I think nutrition in children, even in normal children, is a big deal. And when we think about what children are being fed, and I look at these displaced kids from Syria and elsewhere, and I see them in these camps, and I just think to myself, they're going to inherit these countries. And they're not getting fed, and they've got many other things going on here. I'll just make one other point on this thing, and that is if you go to the World Health Organization, and this has become utter dogma, that if you're going to nutritionally affect the brain, you have to do it in the first 1,000 days, and that is from conception. So that means it's at the end of year 2. Well, that picture I showed you, and I didn't belabor it, but at age 2, your brain is physically pretty much the size of a human brain, but it takes you, then you start laying down all these synapses. It's like that building that's being built somewhere. What do? They pour the concrete, put up the iron and steel, and then they get in there and put the wires in and turn the switches on and so forth. And that's where the cost skyrockets. And the way that it's being approached with these rapid food diets and so forth that are taken to Bangladesh and Malawi is to say, we'll give you these high-calorie diets into year 2. They say goodbye and move on. Well, that's just doesn't make sense with regard to what I know about the biology here. So it's a whole area. Most of the people in St. Louis working on are younger folks that I cheer daily.
Thank you.
Can you please comment on autism, mirror neuron, and any imaging studies done on this?
Say that again.
Can you please comment on autism, mirror neurons, and any image studies on these kids?
I'm not quite sure
Autism.
Oh, autism. I'm sorry. Pardon me for not catching it. I'm almost embarrassed to tell you we have a paper about this, but we thought, and I'm not going to get into the details of that, but because I think when we come to something like autism, it seems to me, as somewhat of an outsider to that whole story, is that there's been one promise after another about its cause. Is it in the cerebellum? Is it this gene? And is it this, that, or the other thing? And they all come on hard times over time. And so I think the tools are getting, from my point of view, and for the kind of tools that we have access to do, and our group writ large is very involved with the people doing the autism work in St. Louis. I think there is a promise for the future here, but I think we ought to be really careful what we're doing and not make promises to an audience that I think deeply wants an answer. So, yes, I've seen data on it. We've published at least a recent paper about some ideas of how to think about it. But is it the answer? I'm quite sure it's not. It's just a teeny step forward.
Thank you, Doctor. What are your thoughts about the attempts to create artificial intelligence and consciousness?
Oh, dear. Would I be unkind to say that is what it's curious that billionaires who've done various things are now very involved in creating funding for this sort of thing, like they want to preserve because only they can afford it. I'm just not very taken by it. I think it's maybe interesting. I don't have a big opinion. It just seems kind of like it's novelty to me, and I'm much more fascinated by how the real thing works.
Thank you.
Okay, last question.
Thank you. I wonder if you could say a little more about the relationship between glycolysis and amyloid plaques, which you showed right at the end of your talk.
Well, there is clearly an association here. How do I want to frame this? These are, well, I'm going to just give you a little history. How do we get into thinking about this? Well, one was the so-called amyloid scans. There they were, and as my friend Randy Buckner said, he could give a talk about the default mode network using PIB scans. It's probably not that clean, but it's very, very suggestive. And so the question is, what does this imply? Well, aerobic glycolysis is very much, well, amyloid is very much involved in synaptic physiology. It's not wild, as far as I can tell, but I could be missing something that is not perfectly defined, but it is. It's part of the normal brain. You were not given amyloid so you could get Alzheimer's disease. It's there for a reason. And also that It's related to activity levels, and this has been demonstrated actually in an experiment we did as a collaboration with Dave Holtzman's lab, who uses dialysis probes for looking at this. And I was curious about his mice, so I called him one day and I said, look, Dave, do your mice, are the plaques uniformly distributed or not? He said, come on over, and he put some slides under the microscope. They're clearly not uniform. So this led to a set of experiments published with a graduate student named Adam Barrow, which showed that aerobic glycolysis and amyloid, soluble amyloid, the stuff that's moving around in there, move up and down with activity. And so that if you wiggle the whiskers, it goes up in localized. And if you shave the whisker, it goes down. So then you use a transgenic mouse, and the plaques in the hemisphere contralateral to the shaved don't develop or have a much retarded growth. So you come around to then a question of activity. Is there something about ongoing activity in the system that is stressing this synaptic physiology, either taking the levels of amyloid abnormally high or holding them just above a certain level over periods of time, and then you eventually get some sort of a precipitation? The cell biology of this, I think, has just barely been looked at. The question of who's playing a role in here, the VIP interneuron, for any of you who know about that little fellow, is a very important player here as a potential mediator of this. It is also true that aerobic glycolysis, I didn't emphasize anything about the details here, but there is a pathway in glucose that goes out for biosynthesis, but is also critical for handling what we call reactive oxygen species. And that involves glycolysis. And so the question is whether in that synaptic physiology we're losing some of the protection that we normally have. And some data, we've been trying to put together a very detailed metabolic lifespan data, and it turns out we have a lot of data, about 160, 170 people. And what was very interesting, and this isn't published, this is just stuff we're working on right now, that your oxygen consumption doesn't really change a whole lot. These are completely normal, PIB negative, all that stuff. It kind of tapers off a little bit. Your glucose consumption goes down much more significantly, and the level of aerobic glycolysis clearly goes down. So the question, are you losing something here in terms of the machinery that protects the brain under these circumstances? I don't know the answer. I'd love to know it. It's very prevalent in my family, and it's a truly fascinating disease problem. But one I think that we're becoming very enamored with looking at, if I will, the tombstones of the disease, but not thinking deeply about this internal biology of the problem. And we had the Kavli put together, I must say, congrats on that one, discussion a couple of weeks ago where a group of people, largely non-Alzheimer's, talked about how you would think about this problem from a scientific point of view. And I found it very stimulating and learned a few facts that one of the things we look at is hyperphosphorylated tau. You've heard of the tangles. Well, it turns out that hyperphosphorylation of tau is a normal phenomenon. And it's most exemplified in hibernation. And it has to do with the retraction of the presynaptic membrane, and then it goes back on again, and you're fine. And so the question is, once again, we need to understand something that we're calling a bad actor. We need to understand what it's doing as part of its daily job.
I'd also like to thank everyone who was here. These were wonderful questions and a good discussion. So thank you. Look forward to seeing you at our next lecture.